Good morning, everybody. This is Kashyap Saharana, your host for today's conference. I warmly welcome all of you to the second day of 4th International Conference on Life and Chemical Sciences, organized by IM Group of Researchers. Today, we go onward on a journey of collaboration and knowledge sharing, bringing together young and passionate researchers from around the globe. Our platform, after 1 year of dedication, has hosted 3 international conferences, 6 webinars. Now we are driving into book chapters and 19 review articles, with 98 authors reflecting our commitment to excellence. And we contributed to social causes like Flood Relief Fund and Palestine Relief Fund. I would like to express my gratitude to the remarkable individuals who have played pivotal roles in our success. Dr. Fahmida Javeen, the Executive Director of and Patron-in-Chief from Bahauddin Zakriya University, Multan, and Dr. Adnan, our Director from University of Swat, who have been actively involved. I extend my deepest appreciation to the Honorable Professor Dr. Memuna Ghafoor Shahid, who serves as patron and co-patron, as well as our esteemed team members Azazul Islam, Mushtaq Ahmad, Miss Faiza, Dr. Sadia Bano, Miss Aksa Iqbal, Safia Khan, Dr. Sara Junaid, Maham Iqbal, Miss Riaj Iqbal, Atir Naim, and myself as well. Together, we lead this dynamic organization with unwavering dedication. Currently, I am doing PhD in chemistry from East China University of Science and Technology, Shanghai. So, as a doctoral researcher myself, I'm excited to be a part of this vibrant conference. I'm very grateful to all of you for joining us today. We will be shortly starting our conference, so if anyone from your friends is missing, please inform them to join quickly as we are all set for the virtual stage for our dynamic and inspiring conference. So let's start with our session with a few verses of Holy Quran. So I would request Mr. Idris Ahmed to please join for the recitation.
I am audible, him?
Yes, you are audible.
Oh, okay. أعوذ بالله من الشيطان الرجيم. بسم الله الرحمن الرحيم. اقرأ باسم ربك الذي خلق. خلق الإنسان من علق. اقرأ وربك الأكرم الذي علم بالقلم. علم الإنسان ما لم يعلم. In the name of Allah, the most gracious, the most merciful. Read with the name of your Lord who created everything. He created man from a clot of blood. Read, and your Lord is the most gracious, who imparted knowledge by means of the pen. He taught man what he did not know. Sadaqallahul Azim.
Jazakallah Idris. Now we will play national anthem.
Thank you, Zora. Moving towards our next guest speaker, Professor Dr. Nazish Mazhar Ali. She has done her PhD in zoology from GC University Lahore and Sylvester Comprehensive Cancer Center, Miller School of Medicine, University of Miami, Florida, USA. Currently, she is working as professor in Department of Zoology, GC University Lahore. Her expertise lies in the field of microbiology, biotechnology, and molecular biology. Welcome, dear ma'am.
Thank you so very much, Miss Safia. I hope you can hear me well.
Yes, ma'am.
Okay, should I share my slide?
Yes, ma'am, if you want.
Okay. All right. Please let me know if you can see the slide.
Yes, ma'am.
Yes, we can see it.
Thank you. Okay. A very good afternoon to our respected researchers, my dear participants. I am Professor Dr. Nazish Mazhar Ali, and the title of my presentation is Incidence of Respiratory Distress Syndrome Associated Pneumonia in Neonates and Role of Pseudomonas aeruginosa in Pneumonia among Infants, Prevention and Control via Biological Antibacterial Tools. Now, about the problem statement or the introduction, the respiratory distress syndrome associated with pneumonia is the most common and serious medical problem that affects neonates in Pakistan. Now, here I would take few minutes to give an entire introduction about my title. Actually, my title comprises of two parts. The first half is based on my review article that is on RDS associated pneumonia. RDS or respiratory distress syndrome, which is actually occurring in neonates, particularly in Pakistan. And the second part is based on my research study that is on the isolation of pathogenic strains of Pseudomonas aeruginosa, which is nowadays being considered as a major culprit in the respiratory infections. And thirdly, we in our research study, we observe the antibacterial effects of some biological tools like plant extracts and green synthesized nanoparticles apart from antibiotics. So let us see about this, because I have actually selected this topic for my presentation because I feel that medical biology or medical microbiology is somewhat being neglected in our country. Most of the research, they go for the research of applied nature, but let me tell you one thing, the biomedical sciences field needs the attention, because when we say the medical microbiology, we say that the doctors, that is the MBBS ones, they should carry out the study. Yes, they do, but they do the case studies because they have their clinical work. It is our responsibility, the researchers, to find out about the infections and the causative agents of the infections. Research work is always done by the researcher, the scientists, not the physicians and the surgeons. So that is why I'm so keen. I'm always so keen, and because of being microbiologist, my special interest is in medical microbiology. I'm always very keen to work, to do research work in the field of medical microbiology. If you see my research articles on ResearchGate, most of them are actually on the infections like eye infections, skin infections, liquor infections, particularly the pulmonary infections. And that is why the reason that this year I wrote a review report, and also I conducted the research study on particularly on the neonates and the infants, because these little ones, these little angels are actually the suffering, they are actually suffering the most from the respiratory infections. So first of all, we must know what is RDS, respiratory distress syndrome. We all know that the human fetus has its incubation period of 9 months, comprising of 3 trimesters, first, second, and third. The first one is basically of organogenesis. The second and the third one comprises of the growth, maturation, differentiation period. And we all know that the lungs are the organs which mature at very end, like at 36 or 37 weeks. So when we say when there is a delivery after 37 weeks, we say it's a full term. But the babies who are born before that time, we call them the premature babies or preterm babies. So what actually happens is that if a baby is born before time, its lungs are not mature, they are not working properly. And why? Because we all know that the gaseous exchange, it actually occurs in the air sacs, in the alveoli, and it is done with the help of a chemical, a substance called surfactant. Now, in premature babies or preterm babies, the surfactant is not being produced, and because of which the little one is struggling to get the oxygen, and because of which we have to put it on oxygen aid and sometime on ventilator. So the reason is that lungs are not working properly and these are not fulfilling the demand of the body for the oxygen. Now, what happens next? Because of the poor hygienic conditions in our hospitals in our country, what actually happens is that the newborn who's already struggling for the oxygen gets an invader in the form of a bacteria or a virus or any other pathogen. This is very unfortunate. Actually, I have witnessed the deaths of many newborns, comprising one of my own baby girls. So it was actually very sad. That is why I have opted this topic. I am so keen to work in that. What is the reason behind that? All the infections that is being occurring in the hospitals, I always say our hospitals, hospital air, hospital environment is the hub of cross infections, nosocomial infections or the notorious pathogens. And what is the reason? Because our hospitals air is not centrally sterilized. Once I was attending a meeting, and we had the chairman of very well reputed funding agency, and I raised the same question that if there is a surgery and we say the surgery is very successful, the patient is fine, but like after a couple of days it develops the infection, and ultimately the patient dies because of the infection although the surgery was successful. And what was the reason? Because any bacteria or virus, it easily invaded in the womb of the patient, and the reason was the contamination. It can be in the air, it can be in the beds, in the wards, in the hands, it can be anywhere. And the reason for that is that our hospital environment is not sterilized. He said that you should go to see the provincial minister and apply your project over there. And I said, sir, she is a minister. She is not a researcher. She's not a scientist. We are. It is our duty to look for the culprits, the microbial culprits, the pathogens who we cannot see, but take their, but they are so potent, then they take even our lives. So that's how it goes. But still, I didn't get any positive response because most of us, particularly in Pakistan, we go for the research of applied nature, and this side is somewhat being neglected. So I'm quite hopeful that I may get some good results to drag the attention of the other participants and the researchers even from our this conference. So there are many reasons, many causes. One, before the birth of the baby, and that is the pregnant women, they can get the infection that can be transmitted to the young one. There is a term amniocentesis, when the amniotic fluid gets low, so we externally add the water in the womb, but that is actually not practiced much here in our country because itself it's very risky, so we avoid that. But still, yes, we have some issues, because if there is a decrease in AFI, we go for early section. And because of early C-section, what actually happens is that the baby is born before time, and when the baby has come from a very sterile environment and suddenly it is exposed to the contaminated environment, and its lungs are not working well, so it's very hard for it to survive. So these are actually the issues that are being there in third world countries, including our sweet homeland. So we must do some precautionary measures. We must find the ways to avoid such conditions. Now, if we see the NMR in Pakistan, Pakistan has a neonatal mortality rate of 41 deaths per 1,000 live births. It's a number. One of the highest in the world, probably the third highest one. This is very sad situation. I have seen parents outside the waiting for their newborns to recover or to die. I mean, a time comes when the parent pray that God, please help our newborn. So this is very sad actually, and the reason is only the unawareness, the unhygienic conditions in our hospitals before the birth and after the birth. It is very important to understand that a newborn that has come from a sterile environment, totally free from germs, we actually have to put it aside and we shouldn't even go near it. What we do actually is that whenever we have a new baby, we just take it, no matter if we are having a flu or we are having a sore throat. It can be a simple flu or sore throat, a pharyngitis or laryngitis for you, but it can be life threatening for the newborn. So we must see for these things that how we should handle the babies. Then even in our nurseries, the conditions are not well. The newborns who are having the RDS, they are kept in the incubators, which are so close to each other. I mean, if one newborn who's having only the RDS and the very next incubator is having the newborn with the pneumonia, I think there are more than 90% chance that the first one will catch the pneumonia because it goes into the air. Your bacteria, your viruses can survive in air. Your bacteria can survive in air for 7 days. And when you have your notorious virulent pathogenic strains, they can survive more. They are very hard. We cannot even imagine. So yes, with the passage of time, yes, we have developed good conditions up to a certain limit in some hospitals. To be very honest with you, we have very good nurseries, but in private hospitals. But if we talk about the conditions in the government hospitals and the nurseries, these are very poor. But you all know that most of the families, they come from the villages or the rural areas and they go directly to the government hospitals, not in the private hospitals. So we are at different levels. We are struggling and we are trying to do some precautionary measures. They are very limited. Like I say here, that the many efforts have been done in our country to improve the public health care, but still we need more and more preventive measures. We do have our limitations. Okay, is there any problem? All right. So yet we have our limitations. That is, we have to develop the good healthcare units, particularly in government hospitals, because the major influx of the patients is from the rural areas. I tell you that if the rural areas patient, they go to the private hospitals, they're immediately being rejected because they do not have the money. And if they do not have the money, they won't get the treatment. Ultimately, they have to go to the government hospitals where there are no incubators, there are no ventilators. And what actually happens is that the newborn who's already suffering from RDS-associated pneumonia get worse. And ultimately, that death is the fate. Every year, the mortality rate of our newborns is 63% to 70%. I mean, this is too much. This is very sad. We all must, I urge all our scientists, particularly the microbiologists, that they should put some attention towards our medical microbiology. Although this field is basically of not applied nature, but the basic nature, but still it is related with the public health. It is related with us. And you know that the human health is actually in turn associated with the economy of the country. If we have good health, if we have good healthcare system, ultimately it will impart good effect on our country and on the economy of our country. So my idea to add this portion in my presentation to just to give you the awareness, especially to our young participants. I know there are so many students and young researchers in our conference. It's kind of maybe a good or novel idea for you all, that if you want to do your MPhil or PhD work, you can actually opt this topic to work particularly in your area, because if I'm living in Lahore, I can collect the data only from Lahore. So the participants who are living in other parts of the country, yes, they can conduct this kind of study in their own areas, in their own hospitals. So ultimately one day we can have a very good data on it. Studies have been carried out by certain physicians or the pediatricians, but that is actually the case studies. They just study the numbers. They are not researchers. They won't go to work on the bacterial or fungal or viral pathogens. That is not their job. This is our job to do that. So hoping to have more and more our young researchers in this field, I would move to the next part of my presentation, that is basically based on my research study. This is actually just recent research study that I supervised, that is of my MPhil student, and I have taken just some part of it because it's a complete study, but I did not take all the data because I was told that you will be getting only 20 minutes. So I just compressed my presentation to wind it up in like 20 minutes. So the second part of my presentation, that is actually the practical work. It was on the RDS associated pneumonia in neonates and other types of pneumonia in infants, and also the role of a very, very notorious pathogen, Pseudomonas aeruginosa. Pseudomonas aeruginosa is a gram-negative bacillus. It is an opportunistic pathogen. And you know, opportunistic word is very dangerous. It's to human or for the microbe. For both the times, it's very, very deadly and dangerous. Pseudomonas aeruginosa respiratory infections are very fatal, very lethal. I have previously conducted the research study on the chronic and acute pulmonary infections. It was actually the thesis of my PhD student. And what we got was very good results on it because it showed so much resistance. Only the marinomes were able to had some impact on it, but still, as the persons vary in human from individual to individual, so the strains in of the same species, they also give a different response to the antibacterial agents. So we actually focused on in this study, we focused on the Pseudomonas aeruginosa isolates. And also we studied, the third part was the use of alternative antibacterial biological tools other than the antibiotics. So basically the objectives of our research work was the screening of Pseudomonas aeruginosa from respiratory tract infections of the neonates and the infants, their identification and characterization. And for that, the first step was the isolation of pathogenic strains, because as I told you earlier, that the good and bad individuals are always there. So in bacteria, yes, you have both the non-virulent and virulent bacterial stains. I'll show you the slides showing the pictures of the hemolysis on the blood agar. Then the assessment of antibiotic resistance or susceptibility of these pathogens. And yes, the evaluation of different plant extracts and green synthesized silver nanoparticles against these bacteria. I just make a scheme of the methodology just to avoid the use of time. So I'm going to summarize it. First of all, the sampling was done. We just took the nasal swabs and the blood samples of the neonates and infants, the infected ones actually, and we spread those on the McConkey agar, because you know that the McConkey agar is a very good medium to isolate the gram-negative bacilli. Then we use citramide agar to isolate the Pseudomonas aeruginosa. We carried out the blood agar test. This is also known as hemolytic assay, in which we see that either the bacteria is pathogenic or non-pathogenic. Bacteria usually show three type of hemolysis, that is the alpha hemolysis, beta hemolysis, and gamma hemolysis. Alpha hemolysis is also known as partial hemolysis. Yes, they are pathogens, but they are not that much virulent. The beta hemolysis shows that the bacteria is highly pathogenic, it's highly virulent. And the third type is the gamma hemolysis, which means no hemolysis. The bacteria just grow on the medium, but it is non-pathogenic. Then we carried out the antibacterial assays that included the antibiotic resistance, the plant extracts activity, and the nanoparticles hybrided with the plant extracts, also known as green synthesized nanoparticles. At last, we actually carried out the ribotyping of our isolated strains just to make sure that this is the strains of the Pseudomonas aeruginosa. First, at the initial step, we did the biochemical characterization, and then at the end, we do the molecular characterization. And in that, we actually do the genomic DNA isolation using phenol chloroform extraction method. It's a very good method actually, and we do it manually. We are not having that much resources, so we cannot afford the kits. So we manually prepare different solutions, and we use them to extract our genomic DNA. Genomic DNA of bacteria is not a very big thing to isolate. You can isolate it well manually. It's of like 12 or 15 kb size, and it can or the scientists, they say you can extract the genomic DNA of the bacteria even using the boiling technique. Well, I have tried all the techniques, and somehow I got the genomic DNA. But one thing is there, the quality of the DNA varies, but of course. And what I have observed using all these methods, that the phenol chloroform extraction method is the best one. Then what we do, we use the universal primers, that is the 27F and 1492R, to amplify our gene, that is a 1500 base pair or 1.5 kb. And then we send this gene product to Korea lab for the sequencing, because we did not have the facility of sequencing in our department. Now, dear participants, you can see over here, this is the blood agar test. All of our 4 or 5 strains, we had multiple strains, but I did not add all the data, so because it's gonna take a lot of time. So I have just put the data of only 5 strains. So you can see over here, right here, that the streak lines, you can see the streaking, these are the bacterial colonies, and you can see the pinkish or clear zone around these colonies. It is actually showing the beta hemolysis. If there was like greenish tinge, then it would be the alpha hemolysis, and only if the colony was grown and no hemolysis, it would have been the gamma hemolysis. But our bacterial strains were the beta hemolytic strains. Now about the antibacterial activity, the third part, we isolated the strains, we did the blood agar test and the other biochemical tests. Then we moved to our antibacterial assays, and the antibacterial assays, first we did the antibiotics, because you all know that whenever we have infection, particularly of bacteria, we use the antibiotics. But let me tell you one thing, the misuse and overuse of antibiotics, including the self medication, has led to the resistance in the behavior of bacteria. They do not respond. I mean, when I was a kid, I was a child, we used to simple the penicillins and other simple drugs, the antibiotics, and we didn't have that much resistance of those bacteria. We never went to IV, the intravenous treatment. But now the bacteria don't agree. They don't agree on the penicillins. Even they don't agree on levofloxacin, ciprofloxacin, the fluoroquinolones. They don't agree on azithromycin, the macrolides. I mean, I wonder that what's going on. The oral dosage has no impact. Then we move to the IM, and now I see that intramuscular is also not that effective. So we have to move on IV. And once we used to take Azomax as OD, single dose, but now it's been given in BD. I mean, you have to take it twice a day sometime, because the infections are not being cured. The days are long. First, if you had like to have take your antibiotic for 3 days, now it's gone to 5, 7, and even more. And what is the reason? Because we do not follow the dosage. We do not follow the timing. If you have to take your medicine after 12 hours, you should set an alarm that after 12 hours you have to take your medicine. And then the doses, if doctors ask you to take the antibiotic for 3 days, you have to take it to 3 days, because even your antibiotic needs to maintain its level in your body, in your blood, to get impact on it. So please be careful while using your antibiotics, and do not go for the self medication. You can ask the doctor. It's not that hard. So what we did actually was we picked some antibiotics and anti-infective drugs. You can see over here we use different types of drugs over here: metronidazole, ciprofloxacin, levofloxacin, and azithromycin. The metronidazole is also known as flagyl. It's very good actually, and even after operations and in infections, it's given IV, and as IV it has good impact, and even as oral drug it's good. But my bacteria didn't bother about it. They showed their sensitivity against other antibiotics, but if you can see over here, even azithromycin, you know it like Azomax, they don't bother it much about it. So, but the levofloxacin and ciprofloxacin have their impact, some impact over here. And these are like only 4 or 5 strains. I mean, you have more dangerous strains. They are wandering in the air or water or food outside in the environment. Let me show you the slide. Yes, over here. We did the antibacterial assays either using agar-well diffusion method or disc plate method. So what actually happens is that we spread our bacteria on the Petri plate, and then we add our antibacterial agent. Here it is the antibiotic, and we incubate it for 24 to 48 hours. So you can see over here the clear zones actually indicate that this antibacterial agent has done its impact. So yes, you see the clear zone over here, and you would be wondering, yes, we can see the clear zones, but these look much, but they are not actually much. So that another option is that we look for, apart from being antibiotics. Now we always say about antibiotics. Antibiotics are one evil used to kill the other evil. And why we say that? Because these have their side effects on your GIT, your gastrointestinal tract, on your liver, on your kidneys. I mean, who knows? I mean, not everybody can know the kinetics and dynamics of the antibiotics. So we just take it. It's a drug. Just take it. We don't know what it is doing inside with the organs of our body. So we have to be very careful. Then we have certain plants. And these plants include the clove, people, aloe vera, onion, neem, eucalyptus, lemon, jamun. And they had their different impacts on these bacterial isolate. Let me show you the slide of the pictures so you can get a better idea. Now you can see over here the clear zones. Now you would be wondering, these clear zones aren't that big. as that of the antibiotics. Yes, my dear participants, you are thinking in the right way. But let me tell you, we are looking for the alternative biological antibacterial agents which actually do not have any or minimum side effects. So if we can go further and farther in this field, we can get more and more good results. And if we are not getting good results only with the plant extracts, then we move to the next step, and that is the green synthesized nanoparticles. People ask, nanoparticles, are their cells poisonous? They can be harmful, but when they are mixed with the plant extracts, their toxicity is being capped. So you might heard this term that this cap nanoparticles, that cap nanoparticles. That is because it improves their efficacy and it reduces their toxicity. So here is a graph. You can see over here we have used the silver nanoparticles with our all the plant extracts, and they have shown variable antibacterial activity. Let me show you the slide of the figures. Yes, here these are some of the green synthesized nanoparticles and plant extracts have shown very good results, I tell you. On one side, if a bacterium is showing resistance to your certain antibacterial drug, and on the other hand, if your plant extract or green synthesized nanoparticle is showing antibacterial activity, it's a very good sign. And that gives us hope that we can extend in this particular area to avoid the misuse or overuse of the antibiotics. Uh, it is actually the slide showing our DNA product and the PCR product. We usually prepare like prepare 0.8% on 1% agarose gel, because the bacterial DNA size is more than 10 kb. So it just stucks, it just comes out the well and it stucks somewhere here. You can see over here on the upside, just near the well. On the right side, we have our PCR product that is a 1.5 kb. And let me give you another thing, and that is if you want to have your genomic DNA run in the agarose, let me tell you, if you do not have DNA ladder, you can run it, because the very first band that comes out the well, it would be your DNA. But for PCR products, especially this advice is for our young molecular biologists, never run your PCR product without DNA ladder. My students have done this mistake. You are wasting the agarose, you are wasting your PCR product, which is very precious, I tell you. So we had like 1.5 kb PCR product. Then we sent our strains for the molecular characterization, and these are the dendrograms and the sequence of our strains. Now, coming to the conclusion that what we actually saw, what we actually did, it's some kind of result and the conclusion. All the strains, they show resistance to metronidazole antibiotic and the antibacterial drug. Now all here means only our strains, like 4 or 5 strains. It's not all that all means all the Pseudomonas aeruginosa from this world. So it's only from our study. All the isolated strains, they were sensitive to ciprofloxacin and levofloxacin. Strain D and E showed resistance against azithromycin, but A, B, C were susceptible to it. Plant extracts of people, eucalyptus and jamun showed the best results. I mean, jamun is very good. We all know that Syzygium cumini. It's a very favorite food. We eat it, and we have heard about its antibacterial, antiseptic effects already. Silver nanoparticles of people, eucalyptus, and jamun showed the best results. Strain C was susceptible to all silver nanoparticles, mixed plant extracts, except the jamun. So now this is very amazing that some antibacterial agent is showing good result on one strain, but on the other side, it is being totally resisted by the other strain. Now, future perspectives, what we should do to treat the pathogenic bacteria other than antibiotics. Yes, we can go with the plant extracts and plant extracts plus nanoparticles. Yes, this is one of our ways that we can do it, but it's not on hospital or commercial level yet, because we all the microbiologists, they are somehow working in their labs on it. Some might be someday we are going to get extraordinary result of it, and then we can provide it in market. Then, to treat the respiratory infections caused by Pseudomonas aeruginosa, this is our suggestion that we can use the silver nanoparticles encapped with different types of the plant extracts. Again, I would like to share that there is very much need of preventive measures than for the treatment. What we do actually, that we let the infection grow inside us. It's growing, and we are saying it's nothing, and when we are about to die, then we go to the hospital, and then your doctor cannot do anything, your hospital cannot do anything. What we have to do, we have to maintain hygienic conditions, not only in our homes, in our hospitals. I have seen people, they are eating over there, they are spreading the garbage all over on the floor of the hospital. For God's sake, that is the place where you go for your treatment. So we must keep it clean. Let me tell you, even there is a dust, even if there is a dust and it is there in your environment, you would be ready to receive so many fungal spores, so many soil borne bacteria in your respiratory tract. Now you know that we have this smoke, and we say that smoke is creating issues in our eyes and in our throat, it is causing infections. Let me tell you, the pollutants do not cause infection. It's your causative agents, it's your microbial agents, your bacteria, your viruses, and your fungi, which cause infections, particularly those who are soil borne. So this lot of digging, lot of construction, all these things are actually resulting in increased respiratory and other infections. And there is a big need to get awareness. We all should arrange certain lectures on the public health and the importance of public health and hygienic conditions. I thank you all for your patience, and I am highly thankful to the organizing committee of the conference, and I wish you all to stay healthy. Thank you so very much.
Thank you very much, ma'am, for sharing your expertise with us. Any query from the audience? Yes, you can ask from ma'am.
Excuse me, ma'am. Why
Yes,
dose is why dose is very important when using antibiotic? It is, I think, highly recommended. I heard it from so many people. So please give me a reasonable explanation behind it.
for what? For what?
Uh, Dose is very important when using antibiotics. Yeah, and
Yes,
whenever we are using antibiotics, so we must have to take care of our dose. Why
yes.
it is highly recommended?
Yes, this is very important. Okay, I have a very simple example of it. I think you're gonna enjoy it. We say, do not slap the kids. Do not beat them again and again. They would become resistant. They would become conditioned. That is why dosage is very important. If one time you slap very lightly, again you slap with a more intensity, and you go on increasing, it would make the kid conditioned to it. It won't affect the kid. Similarly is the case with the microbes, particularly the bacteria. If you would not follow the dosage, the amount, particular amount, and the timing, both things are very important, the quantity and the timing, because your antibacterial agent, your antibiotic, needs to adjust itself into your body, particularly into your bloodstream, because bloodstream is the pathway using which your antibiotic targets the targeted organ. So both things are very important. I say that I have my enemy on the east side, and they are 100 in number, and I would send like 50 soldiers in the west side So would that be effective? No, it won't be effective. So the dosage and the timing of the administration of the antibiotic is very important. That is why I always use the word overuse, underuse, or misuse.
I want to invite Dr. Adnan, who is director of IM Group of Researchers and always being a great sport for us, for his conclusive remarks.
Assalamu alaikum, dear valued guests, participants, and esteemed team. Is the fourth International Conference on Life and Chemical Sciences with the theme From Lab to Life, Emerging Trends in Biological and Chemical Sciences come to an end. I want to express my sincere gratitude and pride for the success of this event. On behalf of the organizing committee, I want to thank our guest speakers, participants, and especially the dedicated team that worked hard to make this conference happen. Our goal focused on providing valuable education and raising awareness through research has come to life through the lively discussion and knowledge sharing during these enlightening days. Organized by the IM group of researchers, this conference provided a common ground for the students, researchers, scientists, and professors in life sciences and chemical sciences to share insights, experiences, and the latest advancement in their fields. The objective we set for this conference were not just met but surpassed thanks to active involvement of everyone present. We successfully encouraged collaboration between experts in biological sciences and chemistry, opening doors to innovative solutions for global challenges, discussions on healthcare, drug discovery, sustainable practices, and the exploration of chemistry, of nature, and biomolecular sciences have undoubtedly advanced scientific understanding, paving the way for future breakthroughs. I want to express my appreciation for the commitment shown by participants in promoting ethical chemical research, environmental awareness, and safety measures. This conference has not only been a platform for intellectual exchange, but also a catalyst for building a community of responsible researchers dedicated to making positive impact on society. Uh, Special thanks to our distinguished guest speakers whose expertise enriched the conference and to every participant who actively contributed to to the discussion. My gratitude also goes to the organizing team whose careful planning and ensure the smooth flow of events. I am confident that the knowledge shared and connection made during this conference will go beyond these walls and make a lasting impact on the laboratories, classrooms, and research institutions represented here. As we part ways, let's carry forward the spirit of collaboration, innovation, and ethical responsibility that defined the fourth International Conference on Life in Chemical Sciences. Thank you all for your unwavering support, and I look forward to future endeavors that continue to push the boundaries of scientific exploration and social impact. Thank you all.